computing, the main task is to find the optimal high-quality solution for the specific problem. In evolution of robots, if both evolution of morphologies and bodies is applied, apart from finding the high-quality controllers for a specific body, we also get a lot of body-brain fitness data points. Inspired by this fact, we wanted to see how we can compare, how we can measure the differences between the bodies and the brains, which one is more important for the evolution. So why is this so important? The main goal of our research is the concept called triangle of life. It is the concept of creating the robotic ecosystem where robots can evolve, learn, and reproduce in their own environment. The concept is a vision with 3 stages: conception, delivery, and fertility. Conception, where we design the genome for the robot. Delivery is the phase where the robot is assembled from the description of the genome. And the fertility is the stage where the robot is ready to create an offspring. And we have 3 processes that lead to each of the stages. Infancy stage is the process that proved to be the most challenging. Why? Because we need to go from the robot that don't have any prior knowledge, and they need to learn and adapt to their own body, even though they don't know exactly how they look and how to control it. In the classical robotics, it's easy because we have a specific fixed morphology. We know how many controllers there are, and it's easy to design the controller. Here, the robot needs to go through the learning loop and to develop it by itself. So that's where our focus is. So yes, like I said, control your own body problem is the biggest challenge, because controller also need to be adapted to arbitrary morphology, thus meaning that algorithm has to be universal that makes this. And controllers may not fit to the different morphologies. Probably they won't. So for each robot, there is specific adaptive process. However, in theory, we could transfer the controllers from the robot to the other, but with a specific design that we'll talk in a minute. So the questions are here. How can we isolate and quantify the effect of the bodies and brains on the quality of the robots? Is there any way that we can sample the differences, that we can say, okay, this is the effect that the body produces, and this is the effect that is caused by the brain of the robot? And then if we can do this, how can we compare them? So how do robots look like? We have this design of modular robots. They are designed by RoboGem project, and we just borrowed the system. It consists, in our case, of 3 different modules. We have the core component, which is in the middle, active joints, and fixed blocks. And this is the example of one robot that we also developed in hardware. However, most of the experiments is done in simulation. So how does the controller look like? In order to make the controller transferable from the robot to robot, we have a fairly complex structure. First part of the controller is morphology driven. It maps the structure of the robot, and we create a CPG network specific for each robot. The second part of the controller is morphologically independent. It consists of CPP network. It is neural network alike with fixed number of inputs and fixed number of outputs, but unlike the neural networks, it also has different sorts of activation functions. It has sinusoidal, sigmoid, linear, etc. So we use these inputs for the inputs of the CPPN, use the coordinates of each neuron in the CPG network, meaning that the coordinate system is the same for every robot. So we just need to determine on which coordinate points we actually have neurons. By doing that, we can, and depending on the complexity of CPPN network, we get the different results, different output results that we set as the weights of the connections in the in the CPG network, and the bias and gain values for each neuron in the CPG. When we set these values, we get different locomotive patterns that the robot produces. So yes, here is the more more descriptive part of the learning process. The controller consists of the topology, that's a CPG, and the weights that we get from the CPP. This part depicts the learning loop for the one robot. So each of the CPP network goes through hyperneat. The hyperneat is evaluated based on the behavior of the robot. And then we select the best CPP networks for the new generation. And gradually we get the better and better robots, better and better locomotive patterns. So idea was, having this in mind, to use different CPP networks, the best one that each robot developed for its own morphology, and swap it in different shapes. We selected 25 robots and that are trained for the locomotive task. We took the best CPP network, best brain they produced, and we swapped to each of them. However, we had two training suits. First one is with the robot, with the initial generation of the robots, and with their best performances. But we did the transferring of the controller to over 10 generations in 3 lineages, having in each generation 20 robots. In total, that is around 600. That is 600 robots, and we selected the 25 best robots out of 600. So we have 2 test suits, and for each of them, we swap the brains and bodies. So this is the tool we used to compare. We call it body-brain matrix, and it consists of 25 by 25 matrix. On the diagonal, we have the fitness for the controller that the specific robot developed. If you have the robot 2, the body 2 of the robot 2 with the brain 2, we got the fitness that is equal to the original test result they granted. And we fill in the matrix with the results they get from combining different brains with the bodies. So this by itself is not useful, but what we can do is we can subtract the original result the robot produced with body-brain pair with the new, with the same body with a different brain, or the same brain with a different body. And we get the positive or negative result based on the gain, based on the difference they produced. Sometimes they can be better, sometimes they can be worse. So if we subtract different bodies, we get the D matrix for the differences in bodies, and if we subtract rows, we get delta matrix for the different brains for the case. And here are the results. Instead of giving the raw numbers, we use the heat map, and we can see that the darker the area is, that the more gain we get from the specific robot. In the 0, 0 corner is going up to diagonally. Those are original pairs. They are neutral, around 0. So what we can see is for the best performing body-brain pair, it also has a lot of benefit when used on different bodies. For the brain is the same. We extracted the different delta matrices, and we have this standard deviation for each row and each column. And we can see here that for fixed bodies, standard deviations for the fixed bodies is lower than for fixed brain. So how do we read this? This means that if we swap the brain in different bodies, it has higher differences than if we swap different brains in the same body. It has lower differences than if we swap the same brain in the different bodies. Meaning that morphology has the higher importance for the locomotive task than the brain. I must say that this only applies for this set of experiments, and it may not be the case in general. We also wanted to see why this happens. As you can see here in group 1, that's the initial group, there is a higher differences than in group 2, where we get the best body-brain pairs over 600 robots. So we use this algorithm called tree added distance that compares the differences between two morphologies. And for group 1, you can see that differences are way higher than for group 2. What this practically means is that after some evolutionary time, robots tend to look alike, so the solutions they produce are likely more to fit to each other. That's why we got the differences in the standard deviations are higher. So in order to answer the questions, first, yes, we can quantify and isolate the effects of bodies and the brains, and the tool that we use is called body-brain matrix. And secondly, how do the effects of bodies and brains compare? In our case, bodies, they have a higher importance than the brains. Thank you. Questions?
So I was wondering, what is the function that in which environment they run the experiments? Because depending on the environment, the movements are more important than the decisions.
So we wanted to minimize the effect of the environment. So we use flat terrain, infinite flat terrain, so they can just develop the higher speed. So we cannot say that because of some obstacles they downgraded the performance or upgraded if they're going down the slope. The simulator kit is called Gazebo. It uses the standard physics model in ODE, and it resembles quite a lot these physical models that we have here.
Were you expecting something different?
Could you repeat the question?
I was just wondering if it was expecting something different, because it's basically testing whether the brain is important for moving rather than the body.
Yes.
So were you expecting something different?
I personally expected to see that brains are more important than bodies.
Why? Because the task is moving, right?
Yes. So if you have good controller, then you can move faster. But it turns out that there is less differences in controllers than in morphologies, and this goes in line with the previous research by Pfeiffer, that where he used the same controller in different morphologies, and you have totally different behavior depending on the structure of the robot. So one robot was jumping in the spot, the other one was crawling, the third one was just spinning the leg, the limb. So,
Okay, I was just trying to compare what you're doing with your life, like how people or animals move depending on the terrain.
Mhm.
If it depends more on the brain than the body. So I was just trying to map real world to what you were doing, if there was something new that we could gain from.
well, this is in line with our research for transferring the knowledge from one robot to the other. So in this way, we can analyze the effect on also on social learning in robots. If we have a good controller, then we can analyze upfront before we transfer it, and we can only we can maybe um, analyze controllers prior we test them.
I have a question about difference from group 1 and group 2.
Mhm.
And it's just not clear for me the difference from groups. How's the population in group 1 and who's the population in group 2 for the subset?
You mean in these two images? Or?
No, that does not. The final results. So you say you had two scenarios,
Yep.
and in one of them the difference is not so great, and the other is reasonable difference. What's different in between the two groups?
The difference is that the first group is from the initial population of randomly generated robots. The second group is the best robots out of 60, and after some time they tend to converge, the
Yes.
morphologies as well as the controllers. So they tend to look alike. The differences are smaller between the body delta and D matrix. And for initial population, the group 1, the differences are more visible.
Okay, but did you think of, because when you think of group 1 and they are just in the initial population, then they're just random, they didn't have the chance to try to fit anything to nothing. And I understand that in the second case they have converged. But did you think of making a comparison more fair, like for instance, maintaining diversity so that they do not converge in the end? So applying anything that would maintain diversity, so they would have the chance of evolving and getting better in locomotion, but also maintaining diversity, even though they would be a little bit worse. And then you could compare, because you would have very different bodies to compare.
Well, it's possible, but in this case, this is the part of the research where we tested the effects of the Lamarckian evolution. So we have this population of 20 robots. They learn how to locomote, they reproduce, and they transfer the best controllers they developed to the next generation. And that's done for the 10 generations. So we had the specific problem that we wanted to test, and out of that, this research derived. But it's possible, and it could be done as a next step. What we wanted to show is that we have this new tool that we didn't have before. Nobody compared the bodies and brains in this way, so it's a good way to compare, but it's also inspiring idea for the future tools that we can develop.
So I have two questions. First one is, I guess to some extent this is dependent on this particular choice of body and brain.
Yep.
Right. So the question is, how would do you have any idea how this would generalize to another setting where you have different types of brains with different types of controllers, or is this a very specific finding for this setup?
I don't think it's a specific. One problem that I see is if you don't have a controller that is transferable to other morphologies,
Of course.
then it's hard to.
Then the question doesn't even make sense, right?
Yeah. So I think it's a good way to compare for any kind of controller that you can transfer from the different.
Okay, so now you're saying, well, the method is transferable.
Yes.
Right? And my question was, would you expect to find similar results or very super different results for different setups? Or no idea?
But I'm not sure. By my opinion, I would hope and expect to get the similar results.
Yeah. And also I was thinking, would it make sense to look at neurobiology? Because natural evolution is a big experiment where animals have changed their morphology under evolutionary pressure, and they have maybe, whether or not they have changed their controller, like their brain structures. Would that make sense as a loose analogy?
Maybe, but it would be interesting to investigate. I'll make a note.
Yeah.